how to determine this table, the Punnett square, dihybrid cross, and knowing the phenotype. If you are going to do the monohybrid cross using this, whatever it is, heterozygous or homozygous dominant, homozygous recessive, then yes, tell me what is the genotype ratio, tell me what is the phenotype ratio for each of those different crosses, okay? For this one, for the dihybrid cross, I will mainly ask you about this, the gamete production. What would you have if you have this parental generation, the F1, and then determine the gamete production for the F2, okay? And then what will be the ratio for that? The F2 generation. So that's what you will tell me is 9 to 2 to 1 ratio, okay? So here 9 double dominant, 3 single dominant, 3 single opposite dominant, and 1 double recessive. Okay. 9 double dominant, yellow and round, and single dominant, yellow, but wrinkle, and here single opposite dominant, that will be the green, right, and the round shape, and here double recessive, that will be the green and the wrinkle.
Can you show how you counted it?
What was that?
How you counted the double dominant.
Yes, what about
How
it?
did you count it?
Oh, how do you count? So yes. So you will have here this one, big Y, big Y, big R, right? Keep doing that, and everything that it has big Y and big R, that is double dominant. Okay? So that will be this one, that will be this one, right? This one gets the big Y, this one gets the big R. Here you will have big Y, big R. Here these are the recessive ones, but it doesn't matter because here are the big ones. So you know that it's going to be both dominant. Here, this one, big Y, so this one will get big Y. This one get big R, big R, big R. And here, this one is going to be big Y right there, right there. And here, this one gets big R. Okay. So pretty much everything that gets big Y and big R, those are your double dominant. So here you have 1, 2, 3, 4, 5, 6. This one gets big Y, 6, 7, 8, and we're missing one. This one right here, 9. Those are your 9 double dominant. Okay. Okay. So that would be right there, double dominant. Okay. Yes. Go ahead.
You
Joe?
gave a pretty bad example.
It's because
Huh?
you gave me an example.
You said like where they are?
Oh no, it didn't make any sense.
Okay. Okay. Single dominant. 3 single dominant. So you can do this one, big Y right there. This one, big Y. Here, this one with little r. This one, little r. And this one, big Y and little r. So these 3 single dominant, right? Only the Y. And this one right here, big R, big R, big R. The opposite single dominant, okay? This one going to be like little y, and this one, single recessive, okay? So, 9, 3, 3, 1. Well, for this one, just know the ratio, 9 to 3 to 1 ratio. Don't, for the recessive ones, once it is skipped, yeah, you will see it in another generation for the recessive alleles. All right, yes, make sure you know how to do that in terms of giving me the genotype ratio, the phenotype ratio, okay? Especially for the monohybrid cross. Dihybrid cross, many things that I want you to know, or that I want to be testing you, is gamete production, right? Whenever you have 2 genes, 4 alleles, what are the possible gamete combinations that you may
What
have?
was that called again?
Gamete production, gametes. gametes, sperm cell generation, egg cell generation. So you have this genotype right here, this is your F1. What possible gametes can you have based from this genotype? So you may have this, you may have that, you may have this, you may have that, right? Those are the possible gamete production based on this genotype.
What word is that after the two?
Alleles,
Oh.
two alleles, two alleles. So here you have two genes, 1, 2, and 4 alleles, 2 and 2. Okay. From this slide, the main thing that I want you to know is here, okay? Law of segregation. What is this? Law of segregation, this table right here? The one with the bad jokes? Tell me the joke. Law of segregation? Hmm? Hmm?
Who's saying the bad jokes?
Right there, they're always laughing.
Not like me, I'm really bad at that.
The timeless inappropriate
The
jokes.
division of meiosis.
Hmm?
It says right here.
What it says right there?
It's really on the spot.
Last segregation, okay? For those two alleles that you have, you only get one. They will separate, right? So you only give one, okay? Right here, these two alleles, last segregation, you only give one. You either give this big Y or you will give this little Y right there, okay? So these are the gamete productions right there. So if you have here this genotype, you can give that, or that, or that, or that. Okay? Last segregation, right here. You have these two alleles, big Y, little y. You can only give one to your offspring. So you will either give the big Y or give the little y. Law of segregation. Okay? Law of independent assortment. This big Y can go either with the big R or it can go with the little r. It doesn't matter, it doesn't care. Okay? It doesn't discriminate. It's not like it's saying, oh, I'm a big Y, I can only hang out with the big R. I don't want that little r.
What kind of like the little thing we did up there?
That's right, that's what it is. That is the law of independent assortment. Okay?
What does 1E stand for? That's
What?
Oh, so the two? Oh.
Yeah, that is one gene, okay? So you have two genes. And here you have two alleles for this gene, two alleles for this gene. So for the dihybrid cross, you're using two genes and 4 alleles. Okay? I think that was one of the questions from the what that is supposed to be. So, but yes, make sure you know that, right? That is going to be on the exam. Definitely, for sure. Okay? Make no mistake about it.
Wait, can you repeat it?
So dihybrid cross, right? Two genes. 1, 2, right? 1, 2, two genes, and 4 alleles. 2 alleles, 2 alleles. So that is 4 alleles.
So this is the genotype ratio. How do we get the phenotype?
This is the phenotype ratio.
Phenotype.
That is phenotype ratio. I will not ask you for the genotype ratio for the dihybrid cross, right? That will be like, tell me like how many of these double Y, double R couples, how many of these, that will be just too much. Here I'm just showing you, I didn't fill everything, right? So you have to do everything. Here you have to put a little y. I didn't put a little y because as long as I have the big Y, I don't care, right? I know there's going to be dominant, right? Same thing over here, right? This little y right there, right? I didn't put it there because I'm just telling you about the phenotype. If I were to ask you about the genotype, then yes, place it there, right? Same thing here, little y, right? Same thing for the little r. I didn't put it there because, you know, I'm just counting for the dominant phenotype, okay? So I'm not going to ask you for the genotype ratio. I only ask you for the phenotype ratio, for the dihybrid cross. Monohybrid cross, yes, I'm going to ask you genotype and phenotype ratio. Okay? Good. All right, so moving on, here, law of independent assortment, law of segregation, and here's clearly depicted right here. This is a good example for that. For those two alleles, you only give one, either the big Y or little y, law of segregation. Law of independent assortment, for this big Y can go with the big R or it can go with the little r. It doesn't matter. Law of independent assortment.
So the segregation one, it discriminates.
Segregation separates, okay? And independent assortment, it can go with either one, okay? Those alleles. It can go with either allele of the other gene, okay? Does that make sense? Yeah? You're looking like, little bit. You want a little example?
Yeah,
Yeah?
example.
Yeah?
Uh-huh.
Okay,
Oh,
again.
done?
Huh?
Some of them participate.
No, there is plenty of more, but I just want to make sure you got this one. You want a little example? Okay, so let's come back again with the example. You two. And you two. Come on. It's going to be really quick. Yeah, you two, come on. Otra vez. So here we have one gene, and here we have another gene, okay? Right there. So we have dominant, recessive. We have dominant, recessive, okay? Yellow color, round shape, okay? Yellow color dominant, here, whenever she's recessive, it is going to be green color, recessive, okay? Whenever she's homozygous recessive. If we have two of her, then that would be green color. But because we have her, then that is yellow color, right? She is being masked, she's being like sequestered, she's not being let express this trait, okay? Same thing over here. Round, dominant, wrinkle, recessive, okay? Because she's here, then the seed shape will be what? Round or wrinkle?
Round.
Round and yellow, yes, right there. Okay, here you have this round and yellow, okay? Now it's going to give the gametes, it's going to produce the sperms, the egg cells, right? To do the F2 generation. So, last segregation, it will only give one allele. From these two alleles that it has for the yellow color or green color, it will only give one. So, okay, last segregation, boom, right here, okay? It will make the sperm, it will make the egg cell. This is the last segregation. That is one egg cell, here another egg cell. It can only give one. Last segregation, separates, okay? Love independent isomer basically means that she can go either with her, both dominant, or it can go with, can you go over here? With her, can you go with that? Recessive, okay? Love independent isomer basically means that it can go with either one of this other gene, okay? Love segregation separates only one allele. Love independent isomer basically how you can arrange. You can arrange with the recessive one or with the dominant one. Same thing for her. The recessive one, she can go with the recessive or she can go with the dominant. It doesn't matter, it doesn't care. Okay? As long as they are not really close to each other on the chromosome, then they will absorb independently. Okay? Far away on the same chromosome or in another chromosome. Does that make sense? Yeah? Okay, thank you. Extra credit points for you. Let's keep moving on because I think we have a lot to cover. So that is the 9:1 ratio, the one that we mentioned, and how you can have some parental types and also recombinant types. The recombinant types, those are the ones that are mixed, that are different from the parental types. Parental types, basically the ones that resemble the parents, right? Parental generation, and the recombinant ones, the ones that are mixed. Okay. Here you have this yellow with wrinkle, and here you have this green with round. Those are the ones that are different from the parental types. Okay. So just know that. Here, what is two genes are on the same chromosome, will they absorb independently? So that basically, if they are really close with one another, then they are known as linked genes. So they will absorb as if they were dependent on one another because they are really close. Okay. So those are the linked genes. If they are far apart on the same chromosome, they will absorb independently due to the crossing over. So if they are like really far apart, the same chromosome, then independent absorption. All right, so here now we go to another subject, okay? So now we're going to go over the Morgan's experiment evidence. This is scientific inquiry. So the first solid evidence associating a specific gene with a specific chromosome came from Thomas Morgan, an embryologist. So he did these experiments on fruit flies, and that provided convincing evidence that chromosomes are on the location of metabolite package. Why? How? Well, he used these fruit flies because they produce many offsprings. Every 2 weeks you will have like some options for 10 generations, and they only have like 4 pairs of chromosomes. So really easy to say, right? Okay, yes, these genes, these heritable factors, they are on these chromosomes, they are located on these chromosomes. So he noted that the wild type, that is a normal phenotype, in this case that will be here, the red eye. That is normal phenotype. That is the wild type. That's what you see coming, okay? And the mutant allele, that will be the one that is alternative to this wild type, okay? So this alternative trait, that will be the mutant allele, okay? That is different from the wild type. So wild type, you have the red eyes, and the mutant allele, that will be the white eyes, okay? So then he did this cross. So here you have the parental generation, you have red eyes and white eyes, F1 generation, all red eyes, and then the F2 generation, 3-to-1 ratio, red eyes and white eyes. Boom. Really similar to the purple and white flowers, right? Right? Except that right here, you are dealing with the sexes as well. These ones are female, these ones are male. Here, all of them, yes, red eyes, but here in the F2, notice that all females, red eyes, but the males, 50 and 50. So now here we're dealing with the sex chromosomes, okay? So notice here what it happens. Here you have the females XX chromosome and the males XY chromosome. Only carries one X chromosome. This is what is known as hemizygous. Only carries one X chromosome. So whatever it has on the X chromosome, that's what you will express on males. Females, you have two X chromosomes, so it depends on the combination of the alleles, what is going to be expressed. So the F1 generation, here you have this gamete formation right there. So this one can only give the X chromosome that is for the wild type, right? So in this case, this X chromosome that will pass to the males, it will be safe because it has the wild type, right? It will be red eyes. And here, it will be passing the Y chromosome right there, okay? So that will be a male, receiving the S chromosome from the mom, wild type red eyes, and the Y chromosome from the dad. So these males, it will be displaying the wild type phenotype, the red eyes. And here, the females, they will express one X chromosome, wild type from mom, and another X chromosome, the one that it has the mutant allele, the white eye, from the dad. But because this is a recessive trait, in other words, it needs to be like homozygous to be expressed, it doesn't express it, because now you have the dominant allele there in the other X chromosome, so they are safe, right? They are heterozygous right there. Okay. So you are going to be seeing the dominant expression, that is the red eye, wild type, okay? So notice that. Mom's, red eyes, wild type. Dad's, white eyes, X chromosome. It has the white eyes, it has the mutant allele. That gives, the dads give the X chromosome to the daughters, right? But they are safe because the other X chromosome, it is from the mom, it is safe, they have the X chromosome, the dominant allele, right? So they are safe carriers, but safe. Yes.
Is that how like you had said a while back, like, oh, it has two X chromosomes, but you don't know which one's from the mom. They give it to male, like the X chromosome from the mom.
It's going like, yes, on the right direction, you have two X chromosomes, one from mom, one from dad, right? In this case, the one from dad, the X chromosome, it carries the mutant allele, but the one from mom, it is normal, it is wild type. So that's what you will express, right? So these daughters, they are safe. These guys, these boys, the sons are safe because they have the X chromosome from mom. Yes.
If there was an F3 generation, would any of the females have it?
Yes, depends on like what is the genetic makeup of that F2 that you produce the F3. Yes.
Okay.
Yeah. And that's the whole point, like that once you have that X chromosome like that carrier, then it will take like the homozygous recessive to be expressed.
Okay.
Yeah. Okay. All right. So now you have here this another. I think I saw another one over there. No. Okay. Somebody wants a question? Okay. F1 generation, right? And then here, what are the gamete production that you can have? Loss segregation, right? So this mom right here can give either the wild type or the mutant allele, right? Loss segregation. You can just give one, either this one or that one right there. Here, the same thing. For the dad, the loss segregation, where you can either give the Y chromosome or here the X chromosome on the sperm, right there. Right? this one is safe. So here, the X chromosome from the dad is safe, but now here, the one from mom is not, but doesn't matter because this X chromosome has a wild type, so these daughters still are safe, right? On the other hand, these boys, these sons, they are not. Here you have the X chromosome from the mom, Y chromosome from the dad, and here this X chromosome with the mutant allele now is going to affect these boys right there because they have the Y chromosome right there. So only one chromosome, only one X chromosome. That's what it takes. This is known as hemizygous because when it only carries one X chromosome, the males, so whatever they get on that X chromosome, that's what is going to be expressed. If you have the mutant allele, then you are doomed. That's it. Here, if you get the mutant allele, well, you still have the other X chromosome. Right? So if you are female, you have both of those X chromosomes. If this one is wild type, then you express a wild type phenotype. So it is not a 3-to-1 ratio as we see with Mendel's purple to white flower. In this case, it is a sex-linked gene. In this case, it is an X chromosome trait that is passed to the offsprings. In this case, they're going to be more affected, the males, because it is passed on the X chromosome. Okay, X-linked gene, X-linked trait that is passed around from the parents to the offsprings. Okay, so in this case, 100% of the daughters, they're good, wild type, and here 50% have the wild type, 50% have the mutant allele of the boys, of the men. Okay, so make sure you know that. Here you have the sex-linked genes. Those are the ones that are passed on either the X or the Y chromosome. And also you will have other genes on the X chromosomes that are not actually related just to like sexual developments. Okay. On the Y chromosome, as that one is smaller, that one has like mostly related like genes regarding like the sexual development, and we also have here the SRY gene on the Y chromosome that goes for this protein that directs development of male anatomical features, like receptor for testosterone, so you can develop the male features. Okay, XY chromosome right there, and females are double X, males XY, and that's how you develop male XX, males XY, females XS, and other animals have different methods of sex determination. These are known as the sex-linked genes. Those are the genes that are located on the sex chromosomes that are passed either on the Y chromosome, Y-linked genes, or on the X chromosome, X-linked genes. The ones who are X-linked genes, they will affect mostly on males. They are passed from males. Okay. And some of them are like also like other genes that are not related to sex, as I mentioned before. And as I mentioned, this one as well, for these X-linked traits, and this one, the males only needs one copy of the whatever they get on the that X chromosome. So those are the hemizygous. And they're going to affect more males than females, as I mentioned before. Right, so going back to that, like how can the females get affected? Well, it depends on the genetic makeup of these parents, right? What is it that they have the genetic makeup? It is what they're going to give to their offspring. So you have here this mom who is wild type for both alleles, right? So here you have, and this dad who is affected, the X has a recessive allele. Then here you have the daughters who are going to be carriers, heterozygous. They are safe because the mom passes the dominant allele. They are carriers because the dad passes the recessive allele. For the boys, they are going to be protected because the mom is going to give the dominant allele, so they are good. Right? The dad is going to pass the Y chromosome. On this one, if the dad is protected wild type dominant allele, but the mom is heterozygous, then you have here the daughters being normal for both alleles, homozygous dominant, but here as well being recessive. I mean being a carrier. One recessive, one dominant, heterozygous. Okay? And that is because they receive this one from the dad and this one from the mom. Heterozygous. Here, on this case, for the boys, they receive the Y chromosome from the dad, and this, the X chromosome from the mom. So one is going to be wild type, dominant, and the other one will be recessive, mutant. Okay, so one is affected. Okay. On this one, if you have heterozygous mom, right, she's playing the normal, wild type, dominant allele, right there, one of them, and the other one is the recessive allele, right, heterozygous. And if that is affected, then now here you have showing that the daughters are 50% heterozygous and 50% affected. Now they're going to have recessive, right? Both of them, homozygous recessive. Because one they get from the dad and one will get from the mom, both recessives. Okay? And here for the guys, for the males, 50% will be wild type, normal, dominant, and 50% will be affected, recessive, mutantly. Okay? So make sure you know these characteristics. If you have this genetic makeup from the parents, what would be the phenotype or genotype ratio for the offspring? Okay? So some disorders caused by recessive alleles on the X chromosome. You have color blindness, you have Duchenne muscular dystrophy that is progressive like muscle degeneration, and hemophilia, the inability or like increased ability to develop blood clot formations. This can lead to excessive bleeding internally or externally. Those are passed on the X chromosomes. So what is the probability that this baby will have hemophilia, X chromosome, recessive? What about if it is a boy or if it is a girl? Okay, so what do we have here? Is this a male or a female? Male. What about this one? Female. So here you have X, and here you have Y. This one is affected, so therefore the X is what? Recessive or dominant? Recessive, right? Remember, these have a hemophilia, X chromosome, X-linked gene, and it's recessive. Okay? And here, this mom is what? You know for sure it has one dominant, right? How do you know if it is both dominant or if it is like one recessive? You look at the offspring, one is affected, so therefore you know that one has to be recessive. Okay? So here you have these guys right here, right? And this one you know that is going to be affected. Because you know this Y chromosome comes from the mom, you know that this S chromosome, which is affected, comes from the mom. This Y chromosome comes from the dad, this X chromosome affected comes from the mom. So by knowing this, that one child is affected, then you know that this is heterozygous. This one is the other one. Okay? So make sure you know that one, okay? Do the monotypic cross where you have here this one, the XN, Y, and that one, the XX, big N, little n. So you give that big N here, little n, here the X N, here the Y, and the Y. Okay? So you have here the males and the females. For the females, what percentage are they affected? 50%. For the males, what percentage is affected?
25.
Males. For the males.
50%?
50, right? These are the males, these two. Right, so this one is dominant, this one is recessive. Okay? Yeah, only that one. Yeah. So this one is Y, this one is Y. Can you see this is confusing? A little bit bigger?
How many percent of the boys is affected?
Hmm?
How many percent of the boys is affected? 30?
The gametes from the mom, the gametes from the dad. Okay? Sperms. So whenever you have here the mom passing the dominant, and here the recessive, and here this one, the Y chromosome, and here this one, the X recessive, X recessive. Okay. For females, what percentage are affected? 50. Here you have females, so 50 are affected. For the males, what percentage are affected?
50%?
50. This 50 right here, this was not affected. Yes, question?
No question.
Jackie? Question?
No, Jackie, Arlen.
I'm confused on why the purple is a little M.
Because it is affected. This purple color is showing you that they are affected.
But wouldn't it be the big N?
Hmm?
Wouldn't it be the big N?
No, it's recessive. It's recessive trait. Okay. So in this case, it is telling you that hemophilia is a recessive trait. So it's a recessive trait, then you know that it's the lowercase. Okay. So that's what it's showing you right there. Okay. That one is recessive, so it's lowercase. Okay. If you were to tell you this one is a disease that is dominant trait, then that would be capital. The ones that are black, the ones that are purple. Because it's telling you that it's a recessive trait, then the ones that are affected, then you know that it's a recessive. Yes?
Kind of like the dominant, if you're dominant, it's like more than likely to happen. They get more than likely to happen.
If it is a dominant trait, yes. If it is a dominant trait, then you have two parents that are affected, one child who is unaffected.
Kind of like with the glasses thing.
Yes, that is if it is a dominant or recessive trait. What those are on autosome chromosomes, okay? Complete dominance, those are in autosome chromosomes. This one is on sex chromosomes, okay? So do not get that confused. The autosome chromosomes, by dominant recessive, these are the X chromosomes. So that means that for males, only one X chromosome is sufficient to develop this trait, okay? For these recessive traits, okay? So, all right, so that's what you have right there. Here, pretty much the same thing right here, right? This is that right there. So you have the XN. Here you have the XX. How do you know if it is heterozygous or homozygous dominant? How do you know if this is capital N or lowercase n?
Because one of them is affected.
Because that one is affected, so therefore you know that it's that, right? So this is what? Affected. What about these two? That would be either this or that. But you know that it's not this because they are not affected, so therefore they must be what? This one. So that they are heterozygous, right? XX and N. So you have this mom, heterozygous, and this dad, who is affected. The baby, what percentage will it be that it will be affected? So that's pretty much this right here. 50% if they are female, 50% if they are male. Okay? Does that make sense? Yeah. Okay. So this is, you are going to be asked to do this, I'm sure you know that. These are sex-linked genes. In this case, they are the X-linked genes. Okay? They're passed on the X chromosome. All right, so females, as you know, they have two X chromosomes. So one of them will be inactivated, condensed. That will be called a bald body. This, for example, the tortoiseshell cats, you can see here these patches of orange and black. That is because on these X chromosomes, one will be inactivated, in this case the one's going for the orange fur, and the one goes for the black fur, that will be activated. Okay? That's what you see there, the black expression. Here for the orange one will be active and the black one will be inactivated. Bald body, condensed. Okay? X chromosome. One will be inactivated. Right, now going to this alteration of the chromosome number for a structure caused by the genetic disorders. So you have these large-scale chromosomal alterations in humans, not in other mouse. They can lead to spontaneous abortions, miscarriages, and or cause like a variety of development disorders based on these alterations on the chromosome number. Okay, plants can tolerate that, so they can have this aneuploidy. These are no number of chromosomes, so they can be able to do that better. So this is a result of non-disjunction. This alteration of the chromosome number is a result of non-disjunction, failure to separate these chromosomes. So they fail to separate then you will have one extra chromosome on a cell, or you will have on the other cell a less chromosome. So you are missing a chromosome. That is non-disjunction. Failure to separate. So for example, if this non-disjunction, whenever you have here the separation of homologous chromosomes, so you will see here, if this non-disjunction, failure to separate of these chromosomes, it happens on meiosis 1, then it will lead to all the daughter cells being abnormal in number. Okay. These two will have an extra chromosome, and these two will be missing a chromosome. Whereas if the non-disjunction happened on meiosis 2, then these two daughter cells will be abnormal, one missing a chromosome, one having an extra chromosome. For these two will be normal. We'll have the normal number of chromosomes. So non-disjunction in meiosis 1, all cells abnormal. Non-disjunction on meiosis 2, two cells abnormal. Okay. And these alterations, right, having an extra number of chromosomes, can lead to like several diseases or syndrome. For example, the trisomy 21, Down syndrome, right? You have like intellectual disability, delayed development, right? Or trisomy 18, Edwards syndrome, as well. You have like intellectual disability, abnormal shape of the brain, fingers, and it can lead to stillbirth or like infant death. Trisomy 13, Down syndrome as well. Same thing, infant death. Okay. So this trisomy, when you have an extra number of chromosomes, can lead to like different conditions, different syndromes. Whenever you have a missing chromosome, then that's what it will lead to a miscarriage, spontaneous abortion. So the embryo will not be viable. The embryo will not be viable. So if it is missing a chromosome, then it will not be viable. It will be a miscarriage.
Would that be the only reason people have miscarriages, just because of the missing chromosome?
There are different reasons why. This is one of
Oh.
them. Yeah, miscarriage don't be spontaneous abortion because they are missing a chromosome. There is only like one exception, that would for the X chromosome. Right? Because you have two X chromosomes, so that would be like one particular exception. So that is the non-disjunction. This is pretty much the same picture, but just here showing you like the sperm cell. So if it happens in meiosis 1, then all cells abnormal. If it happens in meiosis 2, then two cells abnormal. Okay. Aneuploidy, that is the abnormal number of chromosomes. Yes.
So for Down syndrome, if like most cases are like, if they're supposed to be aborted, or stillborn or whatever, how come so many people are stillborn with it?
That's if you're missing a chromosome.
Oh,
That's like leading to miscarriage. Trisomy, that's like having an extra number, then yes, that.
Oh,
can-
that does only to miscarriage.
Yeah, no, it can, yeah. For trisomy 18 and trisomy 13, yes, miscarriage, stillbirth, or infant death.
Okay.
They may be able to live like a couple of months, 9 to 13 months, depends. Trisomy 13 for sure.
Okay, sure.
Right. Trisomy 7, trisomy 18 probably a little bit longer, but yes, those extra number, then yes, it can develop like several complications. Missing a chromosome, then that is yes, like miscarriage.
Okay.
Yeah. Aneuploidy, abnormal number of chromosomes. And you can have also other alterations. For example, deletion, deleting a segment of the chromosome. Duplication, like having a repeat. Inversion, like almost like reversing. And translocation, moving certain segments from one to the other. For example, you can have here. In this chromosome, you delete segment D. So you delete those genes within that segment. Deletion. Duplication, you have here B and C, and then repeat it again, B and C. If you have these, for example, then that can lead, for example, to scleroderma, thickening, because you have like more segments in there, more genes in there, extra proteins in there, and that can lead like to like scleroderma, like thickening the extra collagen on the skin, fibrosis, like that. You can have, for example, inversion. So instead of doing this way, BCD, now it's inverse. Now you have BCB, so it is inverse. And translocation, so you move this end of this chromosome and you put it on this chromosome right here, and this end of this chromosome, you put it back over here. So translocation. So human disorders due to the chromosomal alteration, you have like this, for example, different alterations where you have an extra chromosome, trisomy 21, that's the one that we mentioned. frequency of Down syndrome increases with the age of the mother, a correlation that has not been explained. Many people think because as the age increases, then that failure to separate also increases. So it's unable to separate those chromosomes. That's what you have, for example, right there. Trisomy 21. This chromosome 21 right there, having an extra chromosome right there. And we have other examples. Trisomy 18, trisomy 13, trisomy 7, right? And those clinical conditions. Yes.
At what point?
Yes.
No, you go. Okay. At what point in the pregnancy do people know they have a damaged fetus?
Damaged fetus. Well, like for example, here, like have an extra chromosome.
Yeah.
Yes. So yeah, they can do like different testing. Whenever they're like seeing like something abnormal, like different tests. Yes, you can do that. Like 3 months pregnancy, and then it is something that you can monitor. Right? There are different tests to do that. And for example, like we insert like a probe and extract a little bit of DNA and test. Yeah, you can do that.
Doesn't that harm them?
Hmm?
But doesn't that?
After 3 months, it's better, it's safer. The first 3 months is the most important for development. That's where all the organs are being developed. So yes, you don't want to do that on the first 3 months. After the 3 months.
So you can't really tell if there's something wrong with the kid for like 3 months.
Yeah, I mean, it will take. You can do that for like, for example, like different starting from like the ultrasounds. Ultrasound will tell you, okay, this development. You have the gestational side, you have the jolt side, you have the fetal pole, everything. If you see those, okay, it's developing normal. If you see something that is missing, then yes, probably like, okay, miscarriage, probably because it was missing a chromosome, right? Or something that you're seeing as you're monitoring, right? The placenta, the gestational sac, the jolt sac, then that will tell you, right? If it is going in a normal pattern. After 3 months, then if something of that it seems like abnormal, then yes, time to do the genetic test.
For the extra chromosome, is it always on that one or?
This is the most common one. This is the most common chromosome.
But it can be anywhere.
Yes, yes.
If
And
it's anywhere else, would it be something different?
yes, for example, the one that I mentioned, trisomy 18, that's Edward syndrome. Trisomy 13, that is the Blattau syndrome. Even like trisomy 7. These are more, not as common, but definitely like they are like more detrimental, right? It can lead to like miscarriage or infant death. Like trisomy 13, 7, yes, indeed, yes, infant death.
What about the cat or caterwaul or the cat one?
Oh yes, that's at the end.
Oh,
Yes.
okay.
Yes. All right, so that is for autosome chromosomes, right? But also can be happening on the sex chromosomes, okay? So for example, here you have the Klinefelter syndrome, the non-disjunction failure to separate on the sex chromosome. So you may have double XY, double X, double Y, triple XY. So these are known as the Klinefelter syndrome. This is the XYY syndrome having an extra Y chromosome. Turner syndrome, that is the female that only has one X chromosome. This is the one that I mentioned. This is like the exception that if you're missing a chromosome, then it can lead to a viable embryo because you're missing one X, but you still have another X.
Wait, what about people who are born with both
Both what?
genders?
Chromosomes?
Sure.
or like intersex. Well, yeah, let me touch that on the next slide for the intersex. Well, you have here the trisomy X, like 3 X chromosomes. So if you see that, then for example here, that is 47 having one extra chromosome, they usually have these like male sex organs, usually like small testes, and sterile. Right? This is one of the things that are common whenever you have the non-disjunction on the sex chromosome, sterility. And you might also have like breast enlargement or feminine body characteristics, or you have like double twins. For this one, the 47XYY, double Y chromosome, these one individuals are somewhat taller than average and often have like below normal intelligence. At this time, 1970s, it was thought that these men were likely to be like criminal aggressive, but then that hypothesis was proven over time. And that was because during that time, during this time, they were doing this DNA testing on people who were in jail to exonerate them, right? To determine that they were guilty or not guilty. And this genetic testing, yeah, it was really helping, right? Because many of those times, those people who were thrown into prison, they were thrown into prison because, like witness or something, right? And then were not accurate. So many people were innocent, they were in jail. So they were doing this genetic testing and found that they were innocent, yes. But also, they were determined that many of these men, because most people who are in prison are men, they were determined that they actually had like XYY chromosome. So they were, many people in jail, males, XYY chromosome, men, XYY, jail, aggressive. So maybe this is a trait, a phenotype that you see on this person, XYY, they are really aggressive. But then they did that also on the general population, and they saw that it was that. So that's what it was, I was disproven, right? So yes, on that one, yes, you have the XYY, like normal phenotype, one.
Was it coincidence? Was it just coincidence that they were a bunch of aggressive people?
Well, that was because they were doing in jail, right?
Oh,
so in
okay.
jail, yes, you have like most aggressive people in jail, right?
Do we have to remember anything for the test?
Not like the details, but just like the non-disjunction, right? The name, so mainly this one, right here. Klinefelter, it is having these extra X chromosomes or Y. Right? The XYY syndrome, that, double Y. Turner syndrome, 1X, and trisomy X, 3X chromosomes. This one, you can just learn this basis as this one. This one is just extra information, but mostly the same.
Is
Okay?
that XYY too, right?
What was that? Yes, okay. Then the monosomy X Turner syndrome, that's the one that I mentioned, that is the only viable monosomy in humans, so it will lead to a viable embryo, but 30% of the fetus will die before birth, right? So it can lead to some viable fetus, even though it is missing an X chromosome. That is because you have females that have 2 X chromosomes, so they can actually deal with the missing one. They are genetically female, yes, however, they do not mature sexually during puberty and are sterile, so note that. Non-disjunction sex chromosomes, sterility. Okay. The short stature and normal intelligence. Here, the trisomy X, they are healthy and fertile, and also cannot be distinguished from normal female except by character. So, yes, non-disjunction happens also with sex chromosomes. Yes.
For monosomy X, so it says that they're genetically female, but like, what if they were supposed to be male, but they just didn't get that Y? So is it because like they only have an X, which is why they're genetically female because they lack a Y?
Yeah, and I mean, how would you know that it would have supposed to have an X?
Oh yeah, that's true.
I mean, the Y chromosome.
Right.
Like, what if you have all the male character features?
Okay, now that's a different subject. That goes more to the
But you have
intersex.
one.
Okay. And we have a case study for that. So there is one, and I will show you this more detail. Yes, there is one where you may be XY chromosome, but like phenotypically female, and that is because yes, you have the Y chromosome, but it is like non-functional. The SRY protein is not functional. That is a protein, that is a receptor for the testosterone. So you may have XY and have testosterone, but you do not have the SRY
It's kind of like
gene.
the X is superior.
Okay, now we are going to sex. Yeah, no.
I'm saying like the X and Y, like the Y is not like fully functional.
No,
so it's
let's
like
not go into the
Y is
war of the
like-
sexes, okay?
No.
It's not about being superior or not superior.
Yeah, I was gonna say that, I would have said it.
It is about this, all right? You have the XY chromosome, and not that the X is superior, but it is that the Y is not functional.
So the Y
No,
kinda
you
does
say
more
X
work.
superior.
That's
Is that the
the
Y
does
is
more
not
than-
functional, okay?
That's
That
not
is the
functional,
case.
so
So if
it's
the
not
Y is not functional,
So
then what you
men are
have
not
is the X.
functional.
No, okay, that's another subject. You may ask why is the case that the men are not functional, that they are useless, and that is because of the women's fault. If the women support them, it will be a great man. Happy wife, happy life. Nice
Josh is recording.
Okay. I'm just kidding.
You two go
What-
into
what-
this.
what is
Don't worry,
this?
no one's
You
gonna
know,
see this. No one's gonna see this. Don't worry.
Like, turn off the camera.
Stop recording.
And this is my last time working at UHD. I got I don't know. That was well, what was a joke? I don't remember. Okay, going back.
We'll edit it out.
Yeah.
Yeah.
Like, grab it.
No problem.
Yeah.
Stand by. Alright, so, okay, so the XY, yes, we'll cover that more like in the next, I won't have like case study dealing with that specifically for the non-functional Y function. But same thing for the intercept, right? So you may have like normal development, but something going this array. And then you will have a source of different hormones that are not supposed to be there, okay? And that rush of different hormones are affecting the development, okay? So that's why you may have like seeing this intersect development, okay? So it could be like genetic, yes. It could be hormonal, yes. And that can lead to the different physiology and phenotype, right? And we'll go over more of that in detail. This one is referring more to the non-disjunction, to the failure to separate of those chromosomes, and how can that affect that on the phenotype, right? Somebody asked us the last time about the plan B, okay, so how is this affecting, right? So here is failure to separate those chromosomes. The plan B is dealing with another hormone, right? So that will basically be the low testosterone hormone, and that one, basically what it's doing, it is preventing the fertilization of the sperm and the egg. So it's preventing the fertilization. Not only that, it also is preventing the source of the hormone, the luteinizing hormone, which that one will increase or it will spring the ovulation, ready for fertilization. So this hormone in plan B pill, it is going to block this luteinizing hormone, so it will not be ovulating.
So is that the same thing for birth control, or would it be something different?
This one, it is basically preventing fertilization, right? So it is not that it's killing the embryo, but it's preventing the development of the embryo. So it prevents fertilization, prevents ovulation, and prevents, in case that there is a fertilized egg, it prevents the attachment to the uterus by changing the uterine lining, so it cannot attach, so it's preventing.
But I thought they said you couldn't take Plan B when you're fertile.
So that's another thing. So you take it, and it is like in combination with other hormones, estrogen, that is being caused, or that's being associated, that there has been shown that it's associated as well with like breast cancer development. So yes, I mean, once you are dealing with hormones, then you are dealing with like changing with your normal physiology of how your body deals with response. So, all right, so I think we're getting close to, yes. All right, so let me finish this. Oh, here is basically the gradient chat. For the gradient chat, basically results for the specific deletion chromosome 5. Questions?
Oh yeah, you're saying birth control causes cancer?
So this plan B, it has been shown.
Oh, Plan B.
Yes,
Okay.
Plan B. That this Plan B has been shown whenever it's in combination with other hormones, such as like estrogen, then it has been shown that it has been associated with breast cancer development. Whenever it's in use. Yeah. So mixture of these hormones, and when you use it, yes. Other symptoms, basically you will have a delay on your menstrual cycle, like prolonged bleeding, right? And many like pains with the abdomen and so on. But that's the main one, right? Breast cancer association. Okay. All right. So private chat, deletion in chromosome 5. These children will have this syndrome that they are mentally retarded. They have this cat-like cry, and usually they're like infancy or early childhood. So there's a like deletion of this segment. Okay. So here's now that they're missing the chromosome 5, but here's that they're missing a segment on this chromosome. And then you have here this certain cancer, this chronic myelogenous leukemia, CML, and these are caused by translocation of the chromosome. And that is on this chromosome 9, this end right here, it's going to swap with this chromosome 22. So now you have here the cat from chromosome 22 from chromosome 9, and the cat from chromosome 9 from chromosome 22. This chromosome 22 is known as the Philadelphia chromosome because that's where it was discovered. Okay. And that's it. Any questions? Nothing? Right on time.
So did they just call it cry of the cat because they sound like?
Yes, it sounds like a cat.
I don't know what.
That's in French.